All right, so welcome to the last video in respect to the ion channels subject from our 20th lecture. And we are going to conclude this lecture from what I call attacking the subject from the other end by answering a bunch of questions that was actually phrased from the Department of Biophysics and Cell Biology at the University of Debrecen, which means that now we can look at the questions that they phrased and know what they may ask us in our exam with respect to the subject. So first of all, we're going to cover selectivity of different channels, and we're going to cover how that selectivity is actually obtained. And we're going to talk a little bit about the patch clamp technique. So let's get started. First of all, I'd like to talk about the selectivity of different ion channels. And we need to understand that different ion channels have different selectivity. Some are more selective, and some are less selective. So when we're talking about, let's just start with high selectivity. And these are 4 subunit ion channels. And they're based on specific interactions. And you really need to know how many subunits they're made of. And also what I would do is I'd also make sure that I know an example like potassium ion channels, sodium ion channels, or maybe calcium ion channels or chlorine ion channels. All of these are examples of 4 subunit highly selective ion channels. We also have moderately selective. And this is based on, let's just say, charge-based. So let's just say cation specific. This is just an example, cation specific. So I'm only going to let positive ions in. Regardless of their specific composition of elements. So anything that really is positively charged, I will let in. This is a 5 subunit structure. And a good example for this may be the acetylcholine receptor. Acetylcholine channel receptor. It's a good idea to know. Very good. And the last one would be, well, you wouldn't say non-selective. Well, you can say non-selective, or very low selectivity. This is a 6 subunit channel that basically is size specific. So anything that is that can fit in the size of this pore of this ion channel can actually get through it. And you really need to know how many subunits in each, and give some sort of example. This could be a gap junction channel. It's always good to know at least one example to each in case you're asked. This is really the selectivity, and what different selectivity mechanisms do we have. We have mechanisms based on specific interactions. We have mechanism based on charge, and anything that really would go through it. So let's try and see how these mechanisms work. And here we have an X-ray crystallography depiction of a potassium channel. This is a potassium channel. And what we need to understand about potassium, and if this is the extracellular, this is the intracellular environment. And we have obviously the bilayer here. What we need to understand is that potassium ions are not free-floating in our solution, in our aqueous solution in the body. They're always in a hydrated shell. And we're going to write it here. They're always in their hydrated shell. And that really means is because they're positively charged, they're going to have molecules with partial negative affinity that is going to be attracted to them. And you can see in a aqueous solution how we can find potassium. It's being surrounded by 4 water molecules that have interactions with that potassium ion. And really, when we're thinking about it, we want to have this potassium go through the channel. And we don't really want all these water molecules to come with it. But it is very unfavorable to try and strip these off, because this interaction is almost equivalent as breaking a covalent bond. Breaking this interaction is going to be quite difficult. So how do I get this ion through this channel and into the intracellular environment without breaking these very energetic bonds? How do I do that? Well, it just so happens that what this ion channel does is that it's got some sort of carbonyl oxygens here, 4 pairs, that are facing the inside of the pore. And this is the inside of the pore right here. This is where the ions go through. And if I zoom here, I can see that I have 4 carbonyl oxygens here that are facing the inside, and they're ready to accept this potassium ion as it's going through the pore. And in this sense, they're actually going to strip it of its hydrated shell, and these oxygens are going to function as surrogate water. And this is a really good idea to remember, because they use the same term, surrogate water. So they're basically kind of stripping the hydrated shell without breaking these bonds, and that's how we can make this transition energetically favorable and not invest a lot of energy in having only potassium go through the inside of the pore. And since we're wondering how come only potassium can go through, maybe sodium can go through as well. And the answer is that potassium and sodium have different sizes. Not a very good depiction here, but if this is, for example, potassium, maybe sodium is this big, maybe sodium is this big. And if sodium is smaller, then the distance, I'm going to switch colors here, then the distance between these 4 bonds and these 4 bonds would be different. And maybe it would cause sodium that it tries to go through the pore. Maybe it's only going to cause sodium to interact with only 2 of these oxygen from the surrogate water, and it's still going to be attached to its own water in the hydrated shell, and it wouldn't really be able to make this transition. It wouldn't really be able, because we would still need to essentially break these 2 bonds of 2 of these water molecules that are still attached to it. So it's really not going to be able to get through. And the numbers vary depending on what source you're using. But you can say that around for every 10,000 potassiums that go through, maybe 1 sodium would get through. So this is really highly selective, and it is really a really good filter. And if I had to explain it in an open essay question, what I would say that the mechanism of operation of a highly selective 4 subunit ion channel is basically the presence of 4 surrogate water molecules that can strip this ion off its hydrated shell and just pass it through to the intracellular environment without investing the energy needed to break the bonds of the hydrated shell. And let's see what else we have. We have the cation specific interaction ion channels, and you can pretty much imagine that this is the intracellular environment, this is the extracellular environment. Maybe you already looked at this and you realized, hey, I know how this works. Let's just say I have a positively charged molecule, and you can see that on the outskirts of the pore we have negatively charged macromolecules that are actually going to attract positive charges, and positive charges will be able to go through the pore. Whereas if I had a negatively charged molecule, if it got close to the pore, it is just going to be rejected because negative charges repel from one another. So you can expect all these negative molecules that may be hovering around, they're really going to be bounced off. They wouldn't want to stick around, so they wouldn't be able to go through. And this is really, if I had to answer an open essay question, I would say something along the lines of the semi-selective charge-specific ion channels have negatively charged macromolecules on the outside or the extracellular side of the ion channels next to the pore, and they repel other anions that want to get in the vicinity of the pore. And that is why these anions would not pass through the pore. And if we're looking at the last, I would say, I wouldn't say non-selective, but I would say less selective or hardly selective. So less selective based on based on size, then what we really see is that we have a certain diameter here, and any molecule that answers to that diameter can just go through, just like in gap junction channels. And if you have a molecule that's considerably big, well, obviously it wouldn't be able to get through. So this is really what we're talking about as far as the mechanisms that assure the different selectivities. We're going to keep on going and talk about the patch clamp technique. And the patch clamp is a really good research technique in electrophysiology. And what you really need to know about the patch clamp is this is the patch clamp. You can say that it's in the micrometer scale. Its diameter is, I believe, around 1 micrometer. And again, it depends what sources you're looking at and what different micropipettes you're using. And the micropipette is this body here. And we were using the voltage clamp to kind of study the currents through a membrane. But what we really want to do is study the currents through one specific ion channels, which is a really small body in the micrometer scale. So what we can do is we can attach a micropipette to a cell around in a way that we would cover one ion channel and kind of seclude it from its environment. We can use an electrode here, and this way we can measure the currents going through one ion channel. And this is called a cell attached configuration. Means that I'm attached to a cell. And you can imagine that if I just apply some suction and I suck this ion channel in, I'm going to gain access to the inside of the cell, just like on the right side here. I'm going to gain access to the inside of the cell. And this is called a whole cell configuration. And at this point, you can just imagine I had the ion channel here, and I just sucked it in, and now I have access to the entire cell, or to the entire inside of the cell. And obviously it has more ion channels here, and I can actually measure the currents going through multiple ion channels now. And it's important. I can either use this patch clamp to measure currents going through single ion channels or multiple. This is really a good thing because it allows us a broader spectrum of interest. And other configurations that we may have, if you can imagine that this is the outside of the ion channel, and this is still the outside of the ion channel. What I can do is I can basically take the channel, clamp it, and take it out in a way that I can expose the inside of the ion channel to the outside environment. If it's a little hard for you to look at, let's try this. Try looking at this. I can use this patch clamp here. I can just grab one ion channel. I can grab one ion channel in a way, actually drew it over there, in a way that the inside facing ion channel is actually now in the extracellular environment. This is the extracellular environment. And now this is called the inside out, because the inside of the ion channel is now outside or in the extracellular side. So this is the inside out configuration. And now we also have the outside out configuration where we take it out in a way that its outside stays, the outside of the ion channel still faces the extracellular side as it would in its own cell. You can imagine that maybe the cell looks like this. This is the outside facing ion channel, and this is the inside facing ion channel. And I grab it in a way that still enables the outside to face the outside. And this is the outside out. This is the outside out configuration. And if you're really wondering, what do I need to know about this? Well, the patch clamp, if you're asked to explain what it is, you can just say it's a way of research in electrophysiology. And if you don't remember electrophysiology, you can say that basically you can just attach some sort of clamp with an electrode. And in this way, you can actually measure a single and multiple current from ion channels in different configuration. You can have it attached to a cell in a cell-attached formation. You can suck this one ion channel in, and then you can have access to the whole cell in a whole cell configuration. Or you can do an inside or outside out, outside out, etc., inside out. And what you really want to know is that the way that it differs from the voltage clamp is, if you remember, the voltage clamp we had to insert an electrode inside the cell and then measure it compared to the outside solution. And we can't really insert an electrode to every cell. Some cells don't respond very well to being penetrated by electrodes. So this 1 micrometer scale micropipette can actually allow us to interact with different cells that we can't interact with and we can't research using the voltage clamp technique. What I'd like to do now is I really want to tackle some questions, and that way we'd know maybe what are we going to be asked? What do we have to know? So let's take a look at this, and I encourage you to pause this video now if you'd like to tackle some of these questions. So pause it now if you'd like to work on it yourselves. Very good, so let's get started. What ion channel gating mechanisms are known? And we discussed it in the last video. We have the voltage gated ion channels. We have the ligand gated ion channels. We have the secondary messenger ion channels as well. And we also have the stretching mechanisms, or stretch induced ion channels. And they're all really, we're talking about 4 different mechanisms. And these are all the gating mechanisms that we covered. Next question, what kind of gating states can an ion channel assume? Well, we know that the potassium channel can go from open to closed and back again, and this is for the potassium channel. And for the sodium channel, we have closed to inactive to open back to closed. And this is really what they would like you to tell them. They'd like you to tell them what ion channel does what sort of gating states. And this is this answer. Moving on to the next. By what mechanism structural element is inactivation achieved in certain ion channels? And when they mean certain ion channels, they just don't want to tell you that, oh, there's only sodium that has an inactivation mechanism to it. But really, you were taught, and we were taught, the sodium ion channel has a ball complex, that develops affinity when this channel is conducting. So this is really what they're looking for, the ball complex. And you can actually use this same term because this is what they use, the ball complex. Very good. How can ion channels be classified based on their selectivity? And if you're not sure what we mean, you can just zip on back in this video and look at the highly selective, mildly selective, and you can say either non or less selective, whatever way you want. And this is the way to classify ion channels based on their selectivity. And just a reminder, you have selectivity and gating. Both are the ones that characterize the ion channels. So how is selectivity achieved? Well, basically this was covered in this video as well. We can either achieve it via specific interactions by stripping the hydrated shells from specific ions. We can achieve it by having negatively charged micromolecules on the outside of the ion channel, preventing anions from going in. And we can have just a certain size of pore that would pretty much inhibit bigger molecules from going in. And what is the patch clamp technique? What I would say about the patch clamp technique is that it's a research technique in electrophysiology that allows us to measure currents from either single ion channels or multiple ion channels. What can be studied with this method? Well, the currents through either single or multiple ion channels. And the voltage clamp, we already discussed the voltage clamp, but basically it is a way to measure the membrane potential because it's held at a controlled value independent of different ion currents that go through the channels from different ion channels. And what patch clamp configurations exist? Well, we have the whole cell. We have the whole cell. We have access to the whole cell. We have the cell attached. We have the inside out and the inside out and the outside out. And if you don't remember these, you can just zip on up or rather back in this video and take a look at that. And when we're asking what's the magnitude of the current that is measured by a patch clamp, really I don't imagine that they're going to ask you for current units and specific measurements. They just really want to know that you understand that you can either measure the current going through one single ion channel or multiple ion channels in a whole cell configuration, for example. So this is pretty much it. Hopefully you've gotten some understanding as to the different mechanisms that ion channels are associated with. And I'll see you in the next video.